Welcome back to Sravika's Biology. Today, we're diving into one of the most powerful concepts in cell biology, cell cycle regulation and checkpoints. This topic is not just conceptually beautiful, it's a CSIR-NET favorite, asked again and again in Part B and Part C sections. Let's understand how a cell decides when to divide, when to pause, and when to stop.
Before diving into how the cell cycle is regulated, let's quickly recap the cycle itself. A cell grows in G phase, duplicates its DNA in S phase, and prepares for division in G2. Finally, in the M phase, it divides to form two daughter cells. Notice the checkpoints G, G2, and M, where the cell stops and verifies everything is correct before moving ahead. These checkpoints are the guardians of cell integrity, and they're the stars of our next discussion. Think of cell cycle regulation like traffic lights. Without proper signals, when to go, when to slow, when to stop, chaos occurs. Similarly, if cells divide uncontrollably, they form tumors. That's why regulation is vital. The main regulators are cyclins and cyclin-dependent kinases, CDKs. Cyclins decide when, CDKs perform the action. Together, they form complexes that push the cell into the next phase, but only if conditions are perfect. To make sure everything runs smoothly, the cell has checkpoints, like security guards stationed at critical points. They ensure no damaged or incomplete DNA moves ahead. Cells don't just divide blindly, they cross four gates of control. First, at the G restriction point, the cell decides whether to divide at all. Next, during DNA synthesis, the G2/S checkpoint ensures replication accuracy. Then comes the G2/M, confirming that DNA is intact and ready for division. Finally, the spindle or mitotic checkpoint ensures every chromosome is properly attached before separation. When regulation fails, the result is dangerous, uncontrolled cell growth. Many cancers originate from the loss of checkpoint proteins like p53 or Rb, which normally act as brakes on the cycle. Here's an easy way to remember the major checkpoints. Just think of a race. Ready, set, go, stop. Ready, that's your G restriction point, where the cell commits to divide. Set, the S phase check, making sure DNA is perfectly copied. Go, the G2/M checkpoint, giving the green signal to enter mitosis. And finally, stop, the spindle or mitotic checkpoint, which ensures chromosomes are equally separated. If any of these signals fail, the cell risks mutations or even cancerous growth. Let's quickly summarize. Cell cycle regulation is all about control and perfect timing. The main regulators, cyclins and CDKs, act like the control switches of the entire process. Checkpoints are our safety inspectors, making sure everything happens accurately. And if any checkpoint fails, the result can be disastrous, leading to uncontrolled cell division and cancer. So understanding these regulators isn't just exam smart, it's the key to understanding how life maintains its balance. Alright everyone, that wraps up part 1 of our cell cycle regulation series. Please like, subscribe, and share it with your friends. It really keeps this channel growing.